Hello everyone and welcome back to the channel. In this video, I'll be taking you through the findings from the complete neural mapping of the Drosophila melanogaster brain using a novel electron microscopy technique called TEMCA-2. From this neural mapping, there were mainly 3 insights, but before going over those, let's take a look at the different parts of the fly brain that were involved in this study. Now, this is a diagram of the olfactory neural circuitry of the Drosophila brain. Since this study was mainly focused on the processing of olfactory input, we'll be focusing on this type of specific neural circuitry as opposed to other sensory inputs. Now, the blue region on the right side here represents the antennal lobe, which is the primary olfactory processing center in fruit flies. The AL receives direct input from the olfactory receptor neurons, also known as ORNs, shown here in purple, which detect odors in the environment. Now, each ORN, or olfactory receptor neuron, is specialized to detecting certain odorants, and they're kind of the initial sensory cells that detect the odor molecules. The black pathways here leading from the AL to other parts of the brain are called projection neurons. These neurons carry processed odor information from the AL to 2 primary higher brain centers. This is the mushroom body and the lateral horn. Now, the orange area here represents the mushroom body calyx. This is a region that's kind of associated with learning and memory in insects. Now, the calyx contains Kenyan cells, which are depicted here in yellow, and they receive input from the projection neurons, and they process information related to odors that are kind of just often involved in associative learning, for instance, like associative learning in rewards or punishments, for instance. Okay, since we've kind of gone through the different structures of the fly brain, let's move on to the actual findings of this study. Now, the first finding was with the discovery of the MBCP2 neuron. But before that, since this neuron is located within the mushroom body, let's recap the role of the mushroom body first. Now, the mushroom body was central to the Drosophila learning and memory, but not just the Drosophila, but also all other types of arthropods as well. And it also includes a lot of Kenyan cells, which are integral to sensory input, especially with processing olfactory information. Now, the finding here was that a previously unknown neuron called the MBCP2 that provides multimodal sensory input into the KCs that adds complexity to olfactory and sensory processing was discovered, and this was within the MB and allows for generally a better memory formation. Now, let's see for ourselves how the MBCP2 is connected across the MB regions using this diagram right here. So, this diagram right here, panel A, shows an anatomic reconstruction of the MBCP2 neurons, and it's represented here right here in orange. And this is alongside the whole brain, represented in gray. And also the mushroom body represented in green. And as we can see, the MBCP2 has an extensive connectivity across the whole MB region. Now, to see how the MBCP2 interact with the KCs, let's look at panel 2. Now, in this panel, the MBCP2 neurite, shown here in orange, is situated within the pedunculus, interacting with the KC axons. Now, the arrowheads right here indicate the presynaptic release sites, and they're in contact with the KC axons, which is here in green, which means that the MBCP2 is providing input into these Kenyan cells. Now, this presynaptic arrangement means that the MBCP2 neurons are contributing signals to the KCs in the pedunculus, which is very significant for processing sensory information within the mushroom body. Now, second finding was with clustering of olfactory input. Now, in previous studies, the PNs were thought to connect more randomly with the Kenyan cells. However, this study found out that PNs of similar types tend to just group closely together when they're connected to the KCs in the MB. This process is called tight clustering, and it means that PNs carrying similar types of olfactory information are just bunched together in specific locations as opposed to just being spread out, which was the conventional belief. And why does this matter? This clustering can shape how the brain processes smells, because by having different, similar types of olfactory inputs closely grouped, the brain may emphasize or weight certain smells even more strongly, which could perhaps influence memory formation and decision-making processes in response to these odors. Now, let's look at an application of this sort of finding. If we look at the bottom left model right here, the random null model connectivity, we see that the connections are randomly distributed among the KCs without any specificity, which was the conventional belief. Now, in this model, the food response of PNs, represented here in green, are randomly connected to the P ends, the gray circles, which then connect to the KC dendrites, and there's no really preferential targeting or clustering of inputs into specific P ends. If we look at the bottom right diagram right here, the observed connectivity shows a structured non-random pattern. The food responsive P ends, represented here in green, form targeted connections with the specific KCs, which are represented here in red circles with labeled KC claws and soma, and they're kind of creating a clustered pattern. Now, this diagram suggests that in the Drosophila, food responsive P ends in the mushroom body don't just connect randomly with the Kenyan cells. Instead, they really form targeted over-convergent connections with particular KCs, creating specialized circuits for food-related stimuli. This structured connectivity thus improves the vines' capacity for learning and memory regarding food odors. Third finding was with recurrent microcircuits. Now, while the mushroom body primarily receives input from projection neurons, KCs also form connections within the other neurons within the MB. Now, this study found out that KCs don't just pass information in just a single direction. They create kind of like a sort of a feedback loop by sending outputs to specific selected neuron types within the MB. Now, when the KCs make connections to other types of neurons in the MB, it sets up these recurrent microcircuits, which are essentially microscopic closed loops where signals can cycle through several neurons. Now, the significance of these feedback loops. These loops allow for the MB to refine sensory information through feedback. Now, it's kind of a bit like an echo that allows the brain to hear the sensory input repeatedly, which can help it in refining the sensory detail or even highlighting certain patterns. Now, and this feedback loop may even make the sensory processing even more efficient, which would then in turn improve the fly's ability for even better memory-based decisions. Okay, now let's move on to the actual implications of this study. We now know the specific findings of this discovery, then what does this discovery mean for the greater context of the scientific community? First, as we have established, by providing a full mapping of the neural circuits at a whole brain level, it kind of yields a lot of understanding in how individual neurons may connect with across brain regions. But more specifically, this discovery of the new cell type of MBCP2 and the recurrent microcircuits will allow further into associative learning and memory recall. But secondly, the data set produced from this study was to be released publicly, which means that worldwide, researchers can gain access and utilize these images for comparative studies and such. But thirdly, this innovation in volume EM imaging with the creation of the TEMCA2 system means that EM imaging for model organisms besides the fruit fly may also be possible.